Good afternoon, all. My question is related to nitrosamine group. So what kind of data or supporting document is expected by FDA if the physical standard of NDSRA is not available or cannot be synthesized, or the chemicals, because of the chemical instability, that standard is not stable because of the steric factors. So in such situation, what kind of data you are expected during the ANDA filing? For example, ticagrelol and the NDSRA, if you see, that physical standard is not available because manufacturers cannot able to synthesize that.
Would you mind repeating the question, please?
Mm-hmm. I mean, I can.
Yeah, go ahead.
I can sort of start, okay? But so first of all, if you really cannot manufacture it, you know, so basically, if you can prove that the molecule cannot be formed based upon stress studies, you know, using, you know, various conditions, you know, aqueous conditions and organic conditions, you know, and you can prove that it cannot be formed, then you don't have anything to worry about. It will never exist. So that's one way of doing that. But if it can really form and you can't form that standard, you're going to have to make some assumptions about relative response factors and all that. So there's sort of the two scenarios that would exist. But if your product is if it's very unstable but it exists, it's probably it may not be of tremendous concern, but we would have to see the data and the application to see that.
Just to add to that, ticagrelor does not form, and last year during the meeting, it was discussed that again, the table 1 is a hypothesis. Ticagrelor doesn't form, and I think it's pretty well known. You can probably go with that. I know, yeah, but Andre responded to that, so you have to show it, I guess. I'm so sorry, because ticagrelor was discussed last year a lot. My question is to Dr. Yu. While CPCA has made a huge difference in our lives, but we all know that it's still limited because you know, the nitrosamine, as a student of Dr. Lepke, I can say that after 1990s, very little work got done. Organic functional groups like ether, sulfoxyl groups, even amides esters are not, you know, adequately addressed. Is FDA thinking of doing some studies to like expand the CPCA a little more, add more functional groups? Esters and sulfonils come to mind. I used to work a lot with sulfoxides. They're not carcinogenic, believe me, I'm here. Not dead.
I don't think I'm really capable to answer that question because we have a specific expert working on that questions. I can collect the question and forward to the right person to address it. So,
thank you. I mean, I do agree that CPCA solved a lot of problem, but there's still a lot, you know.
I know they are working on to further refine and based on new information. But again, this is not the area I'm really expert on. And
okay. So, I mean, if you can.
Yeah.
The other question I have is more on the communication.
Time, we do have many questions. Would you mind if we go ahead and allow the other people?
This is a continuation, though. Can I just?
Because we've had these other questions, we'll circle back around if there's time, okay? I appreciate it. Thank you.
Good afternoon. I have a general question regarding the filing categories that are not for NDSRIs that are not on the list. Lately, I've been receiving conflicting information or I guess statuses from the agency. So for example, I have a split of even 50-50 where they reject the PAS and say that it's unreportable, and then another one they say that, okay, it's approved. In all these cases, the commonality was there was no control needed because it didn't exceed 10% of the CPCA limit. So is there a shift here? Because I'm seeing it's putting us in a precarious situation.
Okay, for the supplement, you're talking about the one not on the list or the one on the list table?
Not on the list.
So for the one is not on the list, basically the 26.5 is the conservative one, it's the fundamental one. So if it's less or below 26.5, basically it's CB30. If you go above the 26.5, and then you say, oh, I calculated based on CPCA, that will still be the PS, because that still need a PharmTox to review for the CPCA estimation. But if you get data, your confirmatory data said, okay, I'm less than 10% of the 26.5, then that's an annual reportable.
All right, so it's based on not the CPCA, but the actual value you're getting.
That's true. For these CBE or PAS, it's all based on your confirmatory data. It's not based on your CPCA.
Yeah, I just want to clarify one point here. So if it's published in the list, in our table 1 and or table 2, and you're less than 10%, it's an annual reportable. But if it's a brand new nitrosamine that has never been reported, it's not on our list, okay? And you're proposing a limit that's greater than 26.5 nanograms per day, we need to get an assessment of your CPCA calculation or other limit that you need. And per the guidance, you have to submit that as a prior approval supplement. Does that, so it depends whether it exists, it should be a supplement, okay? Generally, if you're posing greater than 26.5, if it's not on the table, okay? Do you see what I'm saying? But if it's already on the table and you already show that it's below those thresholds by less than 10% or cannot form, then you could be annual reportable.
All right, I appreciate it. Thank you.
Thank you for that. And at this time, we'll go ahead and take some questions from the online in order to balance.
Thank you. This question is directed to Dr. Fu. If a nitrosamine tests negative in the enhanced Ames test, what is the control limit for that nitrosamine? What is the control limit conversely for nitrosamine that tests positive in the EAT? Does FDA communicate externally when it is aware that a nitrosamine poses mutagenic risk?
If I hear the question right, the first one is if it's tested negative.
Yeah.
And can it be controlled?
What is the control limit for that nitrosamine if it tests negative?
Well, it's depend on what is the CPCA, what's recommended CPCA AI limit. That is the default AI limit. So if they want to control above the CPCA-based AI limit, as I mentioned, if only have a negative enhanced Ames assay is not sufficient. FDA require additional 2 tests. It's really depend on this, whether they have what's the established AI limit.
Okay.
The second question,
is what if it what is the control limit? Excuse me, if it tests positive in the AET?
If it tests positive, then it's considered mutagenic, and then we, by default, would recommend based on CPCA-based AI limit if there's no already established AI limit, because sometimes there's established AI limit based on surrogate is kind of acceptable, or maybe some like small molecule nitrosamine, they have a compound-specific AI limit. So that's the case. But if there's no other compound-specific data, then go back to the CPCA-based AI limit.
Okay. And the last component of that, does FDA communicate externally when it is aware that a nitrosamine poses mutagenic risk?
I'm not sure, actually. We generally...
Hold on one second. I think we have an answer here. If you can make your clear for us.
My name is Shruti King. So if there is a mutagenic risk, FDA may let the applicant know that the appropriate control limit is the CPCA-based limit.
Yeah, I thought they have to public, no, that's what I'm not certain.
Yeah. Do you know whether they make it externally to the public?
So if it's a published citation, if NCTR, for example, has identified that positive risk, FDA may refer you to that. Alternatively, if FDA is aware internally of a signal, a positive signal, we may communicate that on an application-specific basis.
Okay, thank you, everyone.
All right, thank you. I think we have a question here in the house, sir, if you'd like to go.
Stephen with a V, last name S-U-R. I'm working for a company based out of the San Francisco area. I think one of the mitigation strategies was adding antioxidant, and I think one of the slides said vitamin C and vitamin E to mitigate the risk of the contamination. Is there specific ones that may be recommended by the FDA, or is it generalized? Is there ones that work better? Thank you.
Yeah, thank you for that question. So as we mentioned in multiple presentations, that it's a fit for purpose. We tried vitamin C. We tried the other antioxidant. Some of them works, some of them doesn't work. So you need to do your method development initially. You need to test them and see, because sometimes they may work initially, but over life cycle, they may not. So you need to test each drug product with the specific antioxidant. But there are a number of those antioxidants were published by FDA that can be used. Thank you.
All right, and it looks like we have another individual stepping up to the mic here in person.
Hi, I was just wondering if the FDA had a progress update on the NDSRI progress reports that were due August 1st, and if and when we can anticipate to receive any feedback on those.
Start. So, we did request that people provide the progress reports on August 1st, 2025. And be honest with you, we are looking at all those progress reports. I can guarantee you that. But, we're going to compile it. We're compiling all the information. And then, based upon that, we will hopefully make a public announcement based upon the next steps.
Okay, so no tentative date yet as to when we can anticipate.
No specific date yet.
Okay, thank you.
Excellent. Then in that case, we'll go ahead and take a few more from online.
Okay, this is for Dr. Kaur. In hypothetical case study 1, why is it necessary to conduct a new bioequivalence study with a new API source, considering that the concern regarding nitrosamines is primarily related to toxicological safety rather than pharmacokinetics or bioequivalence criteria?
Thank you for the question. In the first hypothetical case study, it was not only the nitrosamine issue. So there were CGMP issues also. The deficiencies about the CGMP issues were communicated to the applicant, but they never addressed those deficiencies. Because they never addressed those deficiencies, then the quality of the batches could not be assured. So that's why in that case, the dissolution data only was not enough. If the quality of the bio batch could have been assured, the CGMP issues would have been resolved, then the applicant submitted dissolution data would have been enough, which is the case in most of the cases that when you're changing the API source, all you need is dissolution studies if there are no other issues. Thank you.
Okay.
Okay.
More.
Thank you much for that answer. Next question goes to Dr. Go. The nitrosamine guidance only recommends alternative BE approach for IR product. In case study, it involved a delayed release capsule that was treated similarly to an IR product. Does it mean the agency generally treats DR product as IR product? Thank you.
Yeah, thank you. Thank you for the question. So this is still a unique case. So it doesn't mean we universally treated the DR product as the IR products. And also it's case by case. For example, in this one, it's only formulation change happening in the IR, the sugar sphere. But what if the delayed release enteric layer although the formulation didn't change in that case, if the manufacturing change happened to the delayed release layer, then we would ask for additional data. So it's still case by case. We cannot generalize that.
All right, thank you for that response. Another call for any questions in-house this time. All right, we have another one here at the back mic.
Again, related to nitrosamines. Is strength-based nitrosamine specifications are allowed for same product? Suppose there are 4 strengths in a product, 25 mg, 50 mg, 75 mg, and 100 mg. In any case, strength-based specifications are allowed. Instead of taking directly maximum daily doses, can I use an alternative justification for lower strengths, higher limits?
Oh, I'm sorry.
What's the question? I did not get it. So I did not get. Can you repeat your questions? I did not get what's your question.
Is the strength-based specification is allowed for a product?
Strength-based, how?
Strength.
Like, I know, just it means different. Your one product has different strength, right?
Yes.
So you want to have a different specification limit for each different strength.
Yes.
Normally, we are not allowed because your specification limit is against the API. So it doesn't matter what's your strength, your nitrosamine will still be the same because your API, the strength different is the API content is different for each strength. But you're against the API, so the API will be different and your nitrosamine level will be different because your strength different. But relatively go to the ppm level, they should be all the same. So I don't know why you want to go to, how can you go to different way?
If you see the specification limit calculation, it depends on the AA limit divided by maximum daily doses. That maximum daily doses is of course common for product, but of course if smaller strengths, if clinically not possible for taking that much maximum daily doses, say for example, lower strength only 2 doses is possible, that higher limit is allowed. Of course, recently FDA also accepting with the proper justifications. If at all that is acceptable, why don't you put in some Q&A sessions or in a published guidance so it will be clear to the industry.
So you're talking about like different strengths have different maximum daily dosage. That's really, I did not see much ANDA has a different MDD for different dose. All dose have the same MDD, except I think one ANDA they have from like 5 milligram all the way to 100 milligram. In that case, sometimes the farmhouse allow you using different MDD for different dose when you have a big range. But for majority of drug products, they're not big. It's just like 20, 30, 50. So their MDD will all be the same. So when that case, you will have one value. But when you have a product have a 10 different dose with the MDD from like low to high, then you could be, we do allow you have a different dose, have a different like spec limit when you have a wide variety of the MDDs. But that's the case. It's still case by case. You have to see your product to see if tox allow you have different MDD for different strengths. So that's based on it's nothing to do with strength. It's just for the MDDs. Thanks.
Thank you.
If I could add just one additional piece, so I agree with what Dr. Liang is saying. If there are questions about adjusting the AI limit based on your strength, definitely consider submitting a control correspondence to OGD with that specific question. It is a question that we have addressed directly, so please do that for your application.
Excellent. And I think that seems like all the questions that we have time for. We've reached our time here. So thank you, everybody, for a great conversation with this one.